In this video, we'll be learning about the process or the entire process of glycolysis. Glycolysis is basically the initial sort of reaction that takes place that converts glucose into two molecules of pyruvate. And so to start off, glucose right here is actually a aldehyde open structure, because you can see it's bound to a hydrogen molecule onto another carbon molecule. And so with the enzyme hexokinase, it becomes glucose 6-phosphate. And an easy way to remember this is that hexo refers to the amount of carbons that are in glucose, which are 6, and hexo is a prefix that means 6. Anytime you see the ending kinase, that basically means that a structure is getting phosphorylated, which it is. See, glucose is becoming glucose 6-phosphate. And also, ATP is becoming dephosphorylated into ADP, because that's how it's getting, the glucose is getting the phosphate from the phosphate from ATP. And so after glucose 6-phosphate, phosphohexose isomerase turns glucose 6-phosphate into fructose 6-phosphate. And the main difference here that occurs is that, like I said, glucose is an aldehyde, while fructose is a ketone, because it's got it's attached to 2 carbons, which is what a ketone is. And so isomerase is basically changing the change of an aldehyde to a ketone. So keep that in mind. And so next, fructose 6-phosphate undergoes a change from fructose 6-phosphate to fructose 1,6-bisphosphate via the enzyme phosphofructokinase. And phosphofructokinase, remember, anytime you see kinase, it's adding a phosphate group. And so we go from having just fructose 6-phosphate to fructose 1,6-bisphosphate. So you got a phosphate right here and a phosphate right here. And again, ATP is being converted into ADP, which is where this phosphate is coming from that's being attached here. And so via the enzyme aldolase, fructose 1,6-bisphosphate is converted into dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. But for this enzyme to work, it only works on glyceraldehyde 3-phosphates. And so the enzyme triose phosphate isomerase turns this ketone dihydroxyacetone phosphate into glyceraldehyde 3-phosphate in order for it to be catalyzed by this enzyme, because this is unable to be catalyzed by this enzyme unless it's this structure over here. And so then glyceraldehyde 3-phosphate undergoes an enzymatic reaction via glyceraldehyde 3-phosphate dehydrogenase in order to become 1,3-bisphosphoglycerate. And during that process, a phosphate group is added to 1,3-bisphosphoglycerate, and NAD plus is becoming reduced into NADH. And so then 1,3-bisphosphoglycerate becomes 3-phosphoglycerate via the enzyme phosphoglycerate kinase. But this time, ADP is being phosphorylated into ATP in order to form one of the ATPs that normally is formed during glycolysis. And then we have 3-phosphoglycerate being converted into 2-phosphoglycerate via the enzyme phosphoglycerate mutase. Then from 2-phosphoglycerate into phosphoenolpyruvate, it's being catalyzed by the enzyme enolase. And during this reaction from this to this, a water molecule is actually formed as well. And from phosphoenolpyruvate, it becomes pyruvate via pyruvate kinase. Again, kinase converts ADP, phosphorylates it into ATP. And so at the end of glycolysis, this is the second ATP that forms. So there's a net gain of 2 ATPs, 2 NADHs, and 2 pyruvates. And actually, there are 4 ATP molecules that are formed from glycolysis, but 2 are utilized during these steps over here in order for these 2 structures to obtain the phosphate needed for the enzymatic reaction. And so like I said, the net reaction would be 2 pyruvate, 2 ATP, and 2 NADH. And these are the 3 most important end products. And of course, I mean, there are 2 H2Os and 2 OH and 2 hydrogen ions that are also formed. And so I hope you found this video very helpful in explaining the whole process of glycolysis, and hopefully you've learned some important tips to help you remember the order of the structures and of the enzymes, and which one goes which enzyme goes with which structure. And as always, never stop learning. Peace out.